Hello students, welcome to the program Bachelor of Science, 3rd year, subject Botany, semester 6, course code BOC 108, course title Cytogenetics and Plant Breeding. The title of Unit 4 is Linkage, Crossing Over and Chromosome Mapping. The name of this module is Introduction to Chromosome Mapping and its Application. I am Ms. Marina Janice Rodriguez, Assistant Professor in DCT's Tempe College of Arts and Science, Miramar, Goa. In this module, we will be dealing with the arrangement of genes on chromosomes, major aspects to genetic mapping and its applications. By the end of this module, you will be able to define what chromosome maps are, explain the arrangement of genes on chromosomes, describe the major aspects to genetic mapping, and state the applications of chromosome maps. In daily life, when we go to an unfamiliar place, we make use of maps. Maps help us in spatial thinking. That is, they show us the exact location of a place, and also they show us the location of a place in relation to other places. A similar concept is used in chromosome mapping as well. Chromosome maps show the relative location of genes and other important features based on their linkage. The picture that you see is a representation of a chromosome map showing various genes of Drosophila and their relative location. Arrangement of genes on chromosomes. Now, many genes are located on one chromosome. That is because the number of genes present in an organism usually exceeds the number of chromosomes. The genes are arranged linearly on the chromosome and they behave according to the chromosome during gametogenesis and inheritance. All the genes on a chromosome inherit together and are said to be linked with each other to form what is called as linkage groups. All genes in a given linkage group can be shown in a linear array. A circle or ring exhibits linearity as it is simply a closed line with no ends. A sequence of base pair in DNA is linear. It is appropriate that linkage maps are also linear. Major aspects to genetic mapping. There are three major aspects. First, determination of linkage groups, determination of relative distance between the genes, and determination of gene order. The linkage of genes in a chromosome can be represented in the form of a chromosome map. Let us take the first aspect, that is determination of linkage groups. One has to know that the exact number of chromosomes of that species and then determine the total number of genes by undergoing hybridization experiments between wild and mutant strains. But these techniques can be determined how many phenotypes are linked and consequently their genes during the course of inheritance. The second aspect is determining relative distance between the genes. We have to remember that because of crossing over, not all genes in a linkage group will be inherited together. Recombination tends to occur with increasing frequency. The percentage of recombination can be used to represent a measure of distance between two genes. The percentage of recombination between two loci is directly related to the physical distance between the loci. 1% recombination is equivalent to 1 map unit. We also call it as 1 centimorgan. Map unit is abbreviated or is represented as NU, whereas centimorgans with CM. By analyzing the percent recombination among the progeny of parents that are heterozygous for a number of linked genes, a genetic map can be constructed. For example, two genes that recombine with a frequency of 3.5% are said to be located 3.5 map units apart. The third aspect is determining the gene order. Once we determine the map distance between two genes, it allows us to place the genes in the proper linear order, and hence we determine the gene order on a chromosome. Applications of chromosome mapping. Identification of genes. Chromosome mapping helps us to identify various genes found in plants and animals. These genes may be responsible for specific traits which are beneficial or which may not be beneficial, such as traits responsible for certain diseases. Forensics. Chromosome maps help in identifying potential suspects through DNA evidence collected from the crime scene. On the other hand, DNA evidence also help to absolve those who are innocent. Molecular medicine, gene therapy, is a technique wherein alteration of genes is done so as to suppress a particular disease. It also improves the diagnosis of disease, those diseases which are genetically inherited. Human Genome Project is an international scientific project which aims to map all the human genes on the chromosomes. Agricultural application. Chromosome maps in agricultural applications, they help to produce good crops with good traits, that is, increased production or even nutritive crops. These are my references. Thank you.